Hello, I am Emine Arman Kandirmaz from Marmara University. Today, I want to share with you our experimental work titled The Production of Eco-Friendly Biofilm with Natural Oil for Food Packaging. The use of edible biofilms in food packaging reduced the use of petrochemical polymers that are harmful to human health. The second most common biopolymer in nature, chitosan, is a non-toxic, non-antigenic, biocompatible, and biodegradable polymer. Considering these features, it is frequently used in food packaging applications. Increasing needs for food amount and quality canalize the food industry to fund new packaging techniques that improve storage life and grade of foods. Active packaging systems, one of these methods, can be designed as a sensor, antimicrobial or antimigrant in order to extend the shelf life of the food product and to inform the shelf life of possible degradation. Essential oils, which are antimicrobial, environment-friendly packaging material additives, are used due to their effective biological activities. Essential oils that have known antimicrobial properties include lavender, rosemary, mint, eucalyptus, and geranium. These oils are also edible. In this study, it is aimed to produce antimicrobial, eco-friendly, edible, printable biofilm for active packaging using chitosan and peppermint essential oils. The principal roles of the food packaging are to protect food products from outside influences and damage, to contain food, and to provide consumer with ingredients and nutritional information. The goal of the food packaging is contain food in a cost-effective way that satisfies industry requirements and consumer desires, maintains the food safety, and minimizes the environmental impact. Food packets basically provide barrier and protective functions to protect food against the environmental conditions, fungi, and bacteria. Packaging industry is developing and advancing in order to meet these conditions. These developments are in the direction of being cheap, healthy, biosourced, lightweight, printable, and protective, keeping food fresh for longer. Package should be printable enough to present the final product in well enough. For this purpose, packages play not only a passive promotion role, but also play active roles such as preserving food and extending shelf life in many different areas with newly developed types. Active packaging, the auxiliary packaging materials added to the packaging into the improved use of food. Different agents such as enzymes, natural materials, and bacteriocins can be added into the package, which are used in different materials, papers, films, etc. Some use flavoring agents and important substances for food preservation, such as antioxidants and antimicrobials, can be incorporated into the packaging systems to be released into food products during their storage, instead being used in food formulation as ingredients or additives. This technique allows the release of active substance in correct amount gradually during storage time and in place where more useful. The type of active packaging, antimicrobial packaging, antioxidant packaging, antifungal packaging, oxygen scavenger packaging, ethylene scavenger packaging, and humidity controllers. When it comes to the active packaging, there are many techniques. However, the most common of these can be grouped under 4 items. The first of these is most common packaging material has active properties. In this way, contaminants that can be caused by degradation of food and caused by environment are prevented. In the second type, it is adding active material in a small packet, which is sachet, into a food package. Thus, active degradation products are retained by sachet in possible spoilage and the life of food can be extended. In the third type, the food is directly coated with active material. Thus, it is protected from food spoilage products and its shelf life is extended. However, the biggest disadvantage in this method is chemical migration and chemical accumulation caused by coating material. In the last type, food is placed on a pad containing active material. Active antimicrobial food packaging is an emerging technology in food packaging because consumers are demanding more natural foods with less additive use in food formulations. And antimicrobial active packaging can be achieved by incorporation of antimicrobial agents directly into packaging polymers and by using polymers that are antimicrobial by themselves. The most common antimicrobials are used in packaging natural essential oils, enzymes, silver ions, and organic acids. Peppermint plant is a member of Labitii family and has been frequently used in pharmaceutical industry, science, and science due to the carminative and antiseptic properties. Peppermint oil is effective against many known bacteria and fungi. The sensitivity of mint to gram-negative bacteria is higher than the gram-positive bacteria. Having a high antioxidant effect, peppermint oil can also be used against viruses such as influenza and herpes. In recent years, synthetic polymers have begun to be used more than other materials in food packaging due to the reasons such as ease of production, lightness, cheapness, and accessibility. According to the needs of material to be packaged, the production of wide variety of packaging materials is another factor increasing usage. With the decrease of in oil, which is the main raw material in polymer, the increase in price and the problems getting rid of its waste. The interest in environmentally friendly natural polymer increases. In this respect, interested in natural and biodegradable packaging materials can be used in food packaging is increasing and its production become essential. Natural polymers are described as macromolecules which are completely or partly obtained from renewable plants for plants or animals. Chitosan is a natural polymer that is usually made from shellfish. Chitosan has proven in many studies to have antimicrobial and antibacterial properties. It can be used in active packaging applications due to its ability to mate into film structure and its natural bacterial properties. Chitosan biofilms can be good packaging material due to their low permeability and high barrier against oil, but their low resistance to the water limits their usage areas. In the method, the mean samples obtained from Yalova, Turkey region were using and drying 30 Celsius. These samples were then subjected to hydrodistillation method about 6 hours with Nevo-Kalavenger apparatus. Essential oils were dried with anhydrous sodium sulfate and stored in dark bottles about 4 Celsius. Chitosan blank films were produced with a solvent casting method. A solution of chitosan was prepared by dissolving chitosan in deionized water and acetic acid under stirring 3 hours. After complete dissolution, polyethylene glycol was added a plasticizer to increase film flexibility, decrease fragility, to simplify film separation from Petri. The obtained bio-based polymeric film solution was spilled onto the Petri that about 9 cm diameter and dried vacuum at 30 Celsius for a day. The films were removed from Petri, similar to produce described above, 1 to 0.55 and 10% peppermint oil was added into the same formulation and mixed into ultrasonic mixer about 5 minutes at room temperature, and formulation 1, 2, 3, 4 films were produced. Surface morphology were examined by scanning electron microscope Bio-based films were syro-fractured with liquid nitrogen. Fractured films surfaces coated with gold before some imaging. The transparency of biofilms were determined by Shimatsu ultraviolet spectrophotometer. Antimicrobial properties of obtained films were determined against aureus and E. coli with inhibition zone method. Screen printing was made with processed magenta ink onto obtained films with Arus semi-automatic screen printing machine. Printing parameters were 77 TPC weaving density, 75 degree skew angle, 75 shore hardness. Psi-LAB rates of all films were determined with X-rayed exact spectrophotometer accordance with ISO. The color difference is calculated with delta E 2000 formulation. All calculations were made with 5 repetition. Gloss values measured with Bayaka-Gardner gloss meter according to the ISO. Contact angle and total surface energies of the biofilms were determined with pocket goniometer. As a result, figure shows that polymeric biofilm was a uniformly and non-cracked surfaces. Chitosan-containing biofilms was transparent. The mint oil-containing films were also found to be transparent too. The optical properties of biofilms were investigated with measuring of transmittancy about 300 and 800 nanometer. The transmittance spectrums of biofilms are dedicated in figure, and transmittance value of the films about 750 nanometer and 99 point transmittancy values. This figure was observed that the addition of peppermint oil causes a negligible decreased transmittance of chitosan biofilms. In biofilms include solar chitosan, no respectable inhibition zone was considered versus Escherichia coli or aureus. These results may be concerned the fact that chitosan doesn't diffuse owing to neighboring agar environment in the agar diffusion test method. Thus, only bacteria in direct touch with real medium of chitosan are inhibited. As regards chitosan added peppermint oil, this biofilm demonstrates as an inhibitive impact for 4 concentration. All 2 bacteria type experience as well inhibitory effect, but concentration dependent. From the 2 microorganisms compared, it is understood from the inhibition diameter that the inhibition effect of peppermint oil is more pronounced on aureus. The white substrate was used in the measurement, and the color values of the films are given in table. Color differences were calculated by delta E of films. How it differs from white substrate. When the table is examined, it's concluded but only biofilms shifted the color slightly to yellow, but also biofilms didn't vary the color much. Delta differences didn't noticed with eye because of color differences below 3. It is also within the acceptable references range according to the ISO. When the gloss values were examined, it was determined that all films were at least 5 times glossier than the base paper. This is due to the fact that the rawness of the paper surface is covered by coating and diffuse reflection is prevented. Thus, the papers became glossier. Surface energy plays an important role in interaction of printing process. The content of the substrate is among the parameters that can affect surface, contact angle, and surface energy. The contact angles and surface energies of obtained coatings are given in table. The water contact angle blank formulation was found about 72.3. When the pepper oil added to the base formulation, the contact angle is increased. This is due to the hydrophobic nature of oil. It is seen that the contact angle increases when the amount of oil in biofilm formulation is increased. In all biofilms, contact angle values were higher than blank films. This improves the offset and screen printing printability of biofilms with less ink. Because of oleophilic groups in offset and screen printing, ink structure will interact more with the peppermint oil on the biofilm surface, and good ink surface relation width provided, and the printing process will be easier. Screen printing test prints were made with Arus printing test machine on the all biofilms. Color, gloss, color differences are given in table. When table is examined, it is determined that the color has shifted to the yellow a little, as in unprinted films, it changed the color little. However, the color difference is lower than the films, and it is small to be detected by human eye. As the amount of oil increases, the color differences increases slightly. This shows that the ink tolerates the color differences of magenta tone. When the gloss results were examined, the results obtained in the same direction of the unprinted films. However, it has been determined that due to the solid particle in ink rolling the surface, it has been determined that there is no proportional decrease in glossy in all films, about 1 to 3. As a conclusion, peppermint oil containing chitosan-based biofilms have been successfully prepared. The obtained films have uniform surface and transparent. Biofilms containing peppermint oil have inhibitory properties against both gram-positive and negative bacteria. The highest activity was observed against aureus. When the colors of the films were examined, it was determined that the color differences was below 3, that is, no color differences can be seen by human eye. With increased contact angle of oil-containing biofilms, the surface has become more sensitive to oil-based ink. The printability of the film is increased by adding peppermint oil. Screen prints were made on biofilms without any problem. Thank you for listening my presentation. If you have any question about my subject and would like to ask, you can contact me at my email address.